Good evening, everybody. I'm Margaret Honey, the President and CEO of the New York Hall of Science, and it's my pleasure to welcome all of you to our 5th annual Spring for STEM discussion. Tonight's program, we're really excited about it, features an extraordinary panel of experts who will explore the revolutionary gene editing technology known as CRISPR. But before I get started, I'd like to say a few words about NYSCI and our President's Council. You are scientists, business leaders, entrepreneurs, philanthropists, and others who have an interest in science, in education, and in the cultural and intellectual life of New York and the nation. And you provide crucial support to the museum, for which we're very, very grateful, driving our work as a laboratory for learning, a training ground for young scientists, and an anchor for members of our community. We thank you all who are already members and welcome you who are not yet to learn more and to join us. So I don't need to tell this group what science is and why it's important, but I do want to take a minute to talk about our distinctive approach to science learning and how we help our audiences to experience the real thing, the kind of deep knowledge plus serendipity that Siddhartha Mukherjee calls informed improvisation. We call our approach design, make, play, and we see every day how it enables young people to develop critical thinking, knowledge integration, and innovation skills, and that all-important sort of sense of agency, that sense of I can do this, in ways that are measurable and scalable. Especially today when we are no longer knowledge constrained and when textbooks make a whole lot less sense as teaching tools. Science is most fundamentally about pursuing questions that matter. So we do invite you all to learn more about how we do this. And when you come to NYSCI, we'll tell you about how our museum and our community anchor everything we do. Our work in family and public engagement in STEM, our youth development programming and career ladder, our work with the formal side of STEM education, and of course, our research. We'll tell you about the partnerships that extend our impact locally and around the world, and we'll show you how we do this in the most ethnically diverse county in the country. We are passionate about developing and nurturing the science innovators of the future. So thank you all for being here tonight and joining us on this journey. Now, on tonight's program. We are thrilled to have with us experts and leaders in biology and genetics who will help us understand the extraordinary gene editing technology, CRISPR. Before inviting our panelists to come up to the stage, we're going to play a brief video, so you're going to have to turn that way to see it, that helps to explain kind of the science underlying this technology.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
So I'd like to ask our panelists to come up. So we're joined tonight by Sharon Begley, a senior writer at Stat News. And Sharon is really one of probably the leading journalist in the country that has covered CRISPR and other leading edge science topics. Sharon has graciously agreed to moderate the panel for us tonight, and she has the honor of introducing everyone. So Sharon, I'm going to turn things over to you. Thank you.